Hello. Today I'd like to update you on the progress in bringing the much anticipated technology of multi-column chromatography to use in the manufacture of modern drug therapies. My name is Gerard Gatch, and in the next 10 minutes, I'll describe where in the evolution of our twin column technology we are and what can soon be anticipated. I think you'll enjoy the insights. First, let's review the benefit of twin columns over the conventional single column batch system that have historically been used by our industry. Using two columns in concert allows the continuous feedstock to be purified. Compared to a single column batch configuration, this allows greater manufacturing efficiencies through less resin used, which results in smaller columns and smaller footprint. Faster processing by up to threefold. Less buffers are required, again reducing cost and footprint, and all this adds up to the intensification of downstream process steps. So, what are some of the modes and applications for twin column chromatography? I'll focus on these two configurations, although there are more. Continuous capture, also known as capture SMB, and automated internal recycling using multi-column countercurrent solvent gradient purification, also known as MCSGP. In single column batch capture, a column loading is stopped before the entirety of the bed is used to avoid loss of the product. This results in using the expensive protein A media to perhaps only 60%, leaving much capacity idle. In continuous capture, we use two shorter columns, overloading the first column with no loss of product as it's captured by the second column in series. This is proven to give 2 to 3 higher productivity, greater than 50% better protein A utilization, up to 60% reduced buffer consumption, smaller equipment and buffer tank footprint, and the same configuration allows for poolless integrated polishing. For instance, ion exchange followed by anion exchange with no holdup or break between these operations. Typical applications are for the purification of mAbs, recombinant proteins, vaccines, and other large biomolecules, and the LPLC systems operate at pressures of 2 to 4 bar. Now, the second application for twin columns is for the more difficult to separate solutions where peaks overlap and yields are low, as the operator has to choose to either have high purity, taking a very narrow center cut, or higher yield that incorporates side peaks containing impurities. The MCSGP process, using two columns to continuously recycle impure side peaks to extract the more pure target molecule. So this expands the center cut purification. This results in up to 70% better yield, greater reduced solvent use, up to 60% savings on cost, waste, and footprint. And it decreases manufacturing resources, QC work, and time. The applications for this mode are generally in peptides, oligos, RNA, and DNA molecules, and is part of an HPLC system operating at pressures up to 80 bar. So where in the evolution of this technology are we? Our story starts in 2014, actually a few years prior to that, with the origination of the concepts at bench scale. But let's start with the technology at the instrument scale. In 2014, Chromacon, a Swiss-based company, had patented these concepts and had a working bench scale instrument that was capable of both continuous capture and automated recycling of the side peaks, or MCSGP. It was called Contichrome Cube. But to scale Contichrome Cube to production capacities, a partnership was formed with what was known at that time as Liwa Process Technologies, who specialized in the design and manufacture of GMP scale downstream process skids. Within 2 years, several GMP scale continuous chromatography systems were deployed in the USA and Europe. In the following year, multiple leading manufacturers had used these pilots extensively on monoclonal antibody drugs and duplicated the productivity gains and buffer and resin savings that were seen at the bench scale in their process development labs. Here, I present some of the end-user generated data. Running on a 300-liter bioreactor at 1 gram per liter titer MAB, this user compared in parallel the Conti-Chrome twin pilot to their batch system. Productivity of the Conti-Chrome pilot was measured to be approximately triple the equivalent batch unit and used 30% less buffer. In an extended trial run by BMS, they compared a batch system to a twin system on a 5 gram per liter titer MAB, and additionally looked at the scale down results on a benchtop Chromacon unit and found equivalent performance between the cube and the twin units, as well as a 2x productivity using 50% less protein A and resin and buffers. In the following year, BMS acquired multiple scale-up systems. Thus, from an initial bench evaluation in 2015, by 2019, BMS had begun using these continuous capture systems in clinical manufacturing. A peer-reviewed paper is available that details the successful scale-up work. On this success, other end users have recognized the enormous gains using twin column technology, and now large-scale systems are in GMP production environments in the USA and Europe. Our company has further invested in the technology, building a major research facility in Japan, and funded additional pilot scale systems to further advance the technology. Additionally, a major Europe-based peptide and oligo CMO has ordered 2 full-scale twin column HPLCs incorporating the MCSGP technology. So, these GMP scale MCSGP units will be installed and operating on emerging peptide and oligo-based products in 2021. YMC have also entered into other formal arrangements that are anticipated to result in the full-scale implementation of multiple MCSGP units in Japan and the USA. So in a few years, the technology has evolved from a bench scale proven concept to multiple major installations on GMP products. The journey continues at a quickening pace. Now others see that the significant benefits are being realized by competitors in the industry. So what's next? We'll expand our continuous lab facilities in the US in 2021 to add to the existing labs that are already dedicated to continuous purification in Europe and Asia. We're designing twin column continuous purification systems at double the capacity over the present 20 liter per minute in systems that are currently in GMP production. And we forecast continued expansion in the use of twin column technology at existing end users and new end users. So, to answer the question that was posed at the start of this presentation, where are we today? We've proven the scalability of twin column continuous chromatography. We've successfully commercialized multiple large-scale installations and proven the high productivity of this technology. Thank you for your interest and have a great day.